Okay, Assalamualaikum and a very good day to you. So this is going to be the lecture on glycogen and gluconeogenesis. You should listen or view this lecture after you finish listening to the glycolysis. And as I said before, you can always find these resources online. Okay, for example, from the Khan Academy and so on, which can probably explain to you better in terms of the pathways and also the mechanism of metabolism. Okay. So why glycogen is preferable as energy storage, okay, compared to the fat, all right? So the major advantage of glycogen can produce ATP much faster than fat can produce the energy. And also fat requires oxygen to be produced into ATP, but glycogen can work under the anaerobic respiration or without the oxygen. Okay. However, glycogen can have their own disadvantage. For example, glycogen is much heavier than fat because it can attract 2 molecules of water. Okay, for example, if you have 1 kg of glycogen, the total weight will be 3 kilos, okay, compared to the fat that contain 1 kilogram of energy. Okay, and secondly, fat, it contains a very long carbon which can provide you a lot of energy. Okay, but glycogen is limited. Once your body reach the limit of the glycogen storage, it will start to turning all the glucose into fat. Okay, so basically this is the structure of glycogen. As you can see from here, it is a combination of many glucose molecules that form a mesh network. This one is actually exactly similar to the starch that you can find in the plant. Okay, but this one will reside inside the body, inside the liver, and also inside the muscle. That's why we call them as glycogen. And as you can see from this picture, the black glucose, the one that is being highlighted here, is where the glucose is being turned into a branch. Okay, means that if you see from the chemical standpoint, their bond is a little bit different from the glucose that is being connected without a branch. Okay, so this bond is 1,6 glucosidic bond, while the glucose that is connected away from the branch is 1,4 glycosidic bond. As you can see from here, alpha 1,4 glycosidic bond at the branch and alpha 1,4 glycosidic bond at the non-branch location of the glucose. This will play a significant role in how the glucose can be broken down from glycogen to be changed into ATP later on. Okay, so there are two processes that we will take a look at. The first one is glycogenolysis, means that the formation of glycogen under the excess amount of glucose. And secondly, glycogenolysis is actually the breaking down of glycogen under the lower amount of glucose. And the second one is glycogen synthesis under the excess amount of glucose. There are two major glycogen in our body. The first one is muscle glycogen, means that the glycogen is located at muscle, and the second one is liver glycogen. Both of them contain similar glycogen but different role. Okay? Muscle glycogen, the main role is to provide the energy for the muscle only, but liver glycogen, the major role is to provide the glucose to all the body, all the cells in the body. Okay, so they have a very different role, although they have similar structure. Okay, so we can say that liver is always the mother of everything in the body. It sacrifices itself to make sure all the cells in the body get the enough energy. So when our blood doesn't have enough glucose, liver will be the one that provide the glucose to the blood. Okay, so when the blood contains too much glucose, liver is the one that will uptake more of the glucose through the enzyme we call as glucokinase. Glucokinase is actually the same enzyme as hexokinase in glycolysis in muscle, but their role is different. So liver will be the one that will uptake the glucose and change them into fat to be delivered throughout the body. Okay, and liver also have many other functions, for example, to filter the toxicants, okay, and The major ones is actually to provide or to manage the amount of glucose in the body. Okay, so this is the glycogenolysis, means that the breaking down of glycogen. Okay, it will involve three major enzymes. The first one is glycogen phosphorylase, cut by adding the phosphate. Okay, not the normal hydrolysis. This one is transferase reaction at alpha 1,4 glycosidic bond. Okay, so what happens is this enzyme can cut this glucose at the non-branch location. Okay, it will cut the alpha 1 glucose glycosidic bond, and it will stop until 4 glucose molecule left from the branch. Okay, so this is the branch, right? So it will cut until 1, 2, 3, 4. Okay, sorry, it will cut from here until it reach 1, 2, 3, 4. From the branch, it will stop. Okay, and then the second enzyme will come. The name of the enzyme is glycogen debranching enzyme. Okay, so what happens is this enzyme is another transferase. It will cut this one, the right one, the 3 molecule from the branch, and move this one towards the new non-branch of the glucose. Okay, so this is the second function of the enzyme, and this is the second enzyme, and the second function of the enzyme is to cut the alpha 1,6. Okay, remember just now glycogen phosphorylase cut 1,4, and glycogen debranching enzyme, the first one it cuts the 1,4 as well, but the second one it can cut alpha 1,6 glycosidic bond, which is the second bond that is located at the branch of the glycogen molecule. So it will cut this one, okay, and form one glucose. Okay, now we have a linear structure of the glycogen. Okay, and when you have this linear structure of the glycogen phosphorylase can come again and cut here again until it reach another branch. Okay, until it reach the 4 glucose molecule from the branch. Okay, and then the glycogen debranching enzyme will come. Okay, unfortunately, when the glycogen phosphorylase cut this glucose molecule, instead of producing glucose 6-phosphate, okay, remember, probably I need to, if you don't understand, you can come and see me. Yeah? Usually I need to draw this one. Okay, it will produce glucose 1-phosphate that needs to be converted into glucose 6-phosphate first. Okay, so the third enzyme will come. We call them as phosphoglucometers. As you can see from the name here, the major function of phosphoglucometers is as isomerase. It will convert the glucose 6-phosphate that is been producing here and convert them into glucose 6-phosphate. Okay, you can see here, it produces glucose 1-phosphate here, and the isomerase will convert into glucose 6-phosphate. And this glucose 6-phosphate will enter into the glycolysis pathway. Remember, in the glycolysis pathway, the first one is glucose, second one is glucose 6-phosphate, right? Before it is converted into fructose 6-phosphate, until eventually it will reach the pyruvate. Okay, so what happens? This glucose 6-phosphate can enter into glycolysis and undergo the normal glycolysis. And it is said here that less ATP than normal glycolysis. Why? It means that it requires less ATP than normal glycolysis. Because if you remember, the glycolysis needs to use 2 ATP during the investment phase, okay? From the glucose to glucose 6-phosphate. But in this process, it bypassed the first process. Okay, means rather than 2 ATP, it can only use 1 ATP to conduct the glycolysis. Okay, so in liver, glucose 6-phosphatase is used to form glucose. This one only happens in the liver, but the other things that I mentioned to you before, it happens in the muscle. Okay,